So genes in population. So let's start on the plot on the right. So on the x-axis we have the allele frequency. On the y-axis we have the number of times a certain allele has been observed in our study population, and then the different colors are the different study populations. So this is the graph for the 1,000 Genomes project, which is a project where they studied 2,500 people across the world from, I think, 25 different populations. And then they did, sequence the genome, so they captured most of the genetic variation in their genome. And what this, graph shows is that if we actually the number of, actually on the y-axis it's the number of variants that are specific to a certain population. So if you look on the common alleles, then actually there are very little alleles or a small number of alleles that are only not present in any other population than the one that we are studying. For example, the population from Japan. So almost all common alleles that we see in Japan, we also see in Africa, or we see in Europe. So that's why we can use those genotyping arrays, because all the alleles are common and they can be assessed across the different population. But if we now move the lower frequency, so if we go to this 10%, so 0.1 and below, then you actually see that the number of alleles or variants that are only present in one single population, increases dramatically. So, for example, the African populations that are in the greens. So you see that there are many alleles that are just present in Africa's and nowhere else. Whereas in Europe and Asia, there are also such alleles that are not that common in Europe, but you cannot observe them anywhere in the world. So the take-home message is that if you look at the common alleles, and the common alleles are considered alleles that are allele frequency at least 5% in a population. As I mentioned, almost all the genome-wide association studies and associations with the phenotype are with common alleles. Then actually the most of the genetic variation lies in the allele frequencies between 0.1% and 5%. But this is still the area that is not yet studied or not yet assessed for genetic associations. And here on the right, just like an example of an allele that is only present in very common in Northern Europe, a little bit in Southern Europe, and missing almost everywhere else in the world. So this is the allele that actually codes for the eye color. So the yellow chart of the pie indicates the allele that actually is responsible for blue eyes, and the blue one is for the dark eyes. And you see the frequency is very high in the Northern Europe, and that's also why it's almost the only place you can see people with blue eyes. Although this is not like a rare allele, but the similar plots you can see if you consider most of the rare alleles that you observe in your in your population of interest. And this also complicates your genetic analysis, because if you find an association with a variant that it's rather rare in your population and it's not present in anywhere else, you cannot really replicate your finding, and so it's very hard to say if it's just a false positive association or it's a true through associations. And just a point, rare alleles cluster in families or in closed populations like Finns or Ashkenazis or other population isolates, or then they just appear de novo. So if we now take this genetic information, let's say we have for every individual we have a couple of hundreds, thousands genetic variants assessed, and then we have a wide set of different populations. Can we something say about, can we predict your nationality based on your genetic makeup? And this is a study I did myself back in the days. And if you focus on the plot on the lower left, so there, in the box, in the blue box, here we have Estonians, and there in the color there are the different populations we studied. So here we have Estonians, here we have Latvians, Lithuanians, Russians. Here we have Finns from southern Finland, Finns from northern Finland. Here we have Swedes, Poles, Germany, France, Spain, Italy. So you actually see that it's the genes actually very nicely, reflect, geography where the individuals were collected. And again, why is it so? Because historically, you didn't move hundreds of kilometers to find a mate or your partner, but you just picked a girl from next farm or next village, or if you were extremely adventurous, maybe the next city. But really, like everything, it's the genetic diversity is clustered on those historical populations. And here we only considered the common alleles, so alleles that are present at least in 5% of individuals. But now you can see that if you go into the alleles that are very rare within a population, actually you can draw very precise maps of populations and their differences. And then what was surprising, this is Estonia, very small. We have only 1.2 million Estonians living on this chunk of land. But if in the biobank we knew the birthplace for every participant, and if I just this is as you mentioned, it's a typical this principal component analysis or multiple dimensional scaling analysis on the genetic data, and those are just the two first axes that are plotted here. So here, if I took the average for a certain for a given county where people were born, and then again made this plot and overlaid this with the geography of Estonia, then actually the geographic position of a county and the genetic positioning of the county actually is exactly the same, although every county is actually 25. It's I think 40 times 40 kilometers is this the average is county in Estonia, and you can predict in a very certain accuracy that you are either from a southern Europe, southern Estonia, or northern, or in the northeastern or northwestern. So it's quite powerful method. And then again, why genes mirror the geography? If you again look into the European scale, then actually there are the 5 different main drivers. So it's the height, then it's the skin color, it's the eye color, it's your immune system, because the bugs are different in north than in south, and then also actually ability to drink milk. So those are actually the 5 factors that drive this geographical clustering of the different populations. And as I said, it's quite surprising that we are able to see this kind of substructures within Estonia, which is a very small country on the population size. But again, why this is happening is that, for 800 years the Estonians were serfs for the landlords, so they couldn't really move freely around, and so the alleles start to again like aggregate, or certain alleles are more frequent in one county or one region than in another. And then actually, if you think, actually it's almost like everywhere, maybe less in US, because there has been a lot of like migration and lots of waves, people coming in, and lots of mixing different not ethnic backgrounds, but this national backgrounds. But in the more endogamous populations, actually what is like, at some point, every Estonian is related to every other Estonian. It's assumed as longer back you go in the history. Like maybe I'm related with Uku sitting there, and if I go back like 15 generations, what this means, like in the 14th century. But but it also depends, because for example, I'm here, this bottom black dot, but if you go back into the tree, you start to have many ancestors, and there can be like a place where we with Uku converge. And for example, may have a mutation, and then by chance Uku just has to like got the same region from his ancestor, and he may also have the mutation. So those are the things that one can do in studying special populations, and at some point in any population. So there are some important statistics or tendencies in the genetic allele frequencies in a population. So one of the things that is very handy is the Weinberg equilibrium. So this is allele and genotype frequencies in a population will remain constant from generation to generation in the absence of evolutionary influences. And what does it mean? That if you basically you take the square your one allele frequency and the other allele frequency, and the middle is 2 times one allele times the other allele frequency, this has to be 1, and then it doesn't matter in which generation you look at it, it always has to sum 1. So if allele frequency for one allele goes higher, the other ones has to automatically go lower. But this is very handy, especially when the QC issues that David was discussing yesterday. So if your allele or genotype frequencies, or your allele frequency for a certain genotype, that you see a awesome p-value with your trait is way out of the equilibrium, you can use Pearson chi-square test to estimate if the observed frequency or genotype count is significantly different from the expected. And if this is indeed the case, then it's not usually that you have some evolutionary processes ongoing, but basically it just indicates that the genotyping technology, or the way how we assess the genotype is not okay. So this is actually one of the main tests if you are having if you have access to genotype data and you are running an association. So Hardy-Weinberg equilibrium is definitely something you should assess for every variant that you are testing, or at least for those that you see significant associations, because it can be that in a genotyping technology, for some reason, one of the genotypes systematically doesn't get detected, and this is why you think that there is an association, but actually you're just having non-random missing data. So this is one of the principles in population genetics that is helpful to know and understand. Then I think what other aspect I think we have to spend some time on it already, because some confusion is it's called like linkage disequilibrium, and this comes from this Mendel's laws and how the alleles and loci are linked in the chromosome. So if linkage disequilibrium is a non-random association of alleles at two or more loci at descent from a single ancestral chromosome. Which means like if linkage equilibrium is that that all the alleles segregate independently in the population, as was the Mendel's second law. But for example, if we have two genes that are next to each other in the chromosome, and they are so close to each other that the crossing over events do not happen, or if it happens very rarely, it means that those two genes or two alleles always go together in the genome or in a population. And this is just how to, and for this linkage disequilibrium, the genome, this 3.3 million or billion base pair, can be roughly divided into 1 million this kind of blocks, what we call haploblocks. And those 1 million haploblocks are independent. And there was a question like, how we define this independence. Is it like a distance, like a physical distance, or is this genetic distance, like the centimorgans? So it's actually, it's both. Sometimes, two blocks which are, let's say within 5,000 or 50,000 base pairs are completely independent, and sometimes there is a block of, let's say, more than 6 million base pairs that is completely linked, so always goes together. So it again depends on those crossing over hotspots or a combination hotspots. How it it's just like two graphs that I think somehow help to understand how those blocks form within a family or within a population. So in the left we have the family, so it's like the mother's chromosome and father's, and this with the red stripe is where the recombination happens. There it's like a full chromosome. Now it gets like two parts, like from the mother's first chromosome and father's first chromosome, and then the next generation there is like a new chromosome introduced, and there is a new crossing over, and then the original ancestral blue chromosome is only like 1/3, and then in the next generation yet another new chromosome gets introduced, another crossing over happens, and so on and so forth. And so over, in this case, 3 generations, from the 2 original ancestral chromosomes in the family, is only left, 2 small chunks. And for example, if a certain mutation runs in the family, and you have like all the third generation offsprings genotyped, and only those that have a disease who have this ancestral parental chromosome chunk, then you can say, oh, this causal mutation lies somewhere in this blue area. But the same thing is true in the population, because there we have a initial generation, and there is a certain number of chromosomes, and those have like a certain number of chromosome, like these haplotype blocks that are in varying lengths. And again, like over the generations, and here the generations are much larger, like the steps, like 100 generations or 1,000 generations, And again, because of those, crossing over events that happen randomly over all the chromosomes and at every meiosis, and over the generations, the haploblocks, the initial haploblocks or founding haploblocks actually gets smaller and smaller and smaller and smaller. And then, what this was, initially is like only small pieces after 1,000 generations. And that's why, some of the haploblocks can be like wide. Let's say, I think the usual haploblock length, it's like, I think if you usually like you have two associations and you want to know if they are independent, then you go usually like half a megabase, but sometimes like 100,000 base pairs is enough. But what is the take-home message is that although there are like this crossing over events and we roughly have 1 million independent blocks in the genome, and for every block there are several versions of it. But if within this specific block, all the variants or all the alleles or all the genes are linked. So now if we go and then if we can identify this particular haplotype, then we can actually automatically say what are the other variants or other mutations within this haploblock. And this is this linkage disequilibrium. This that the that the variants are linked across over a certain length, physical length. This enables you to not work with 3.3 billion base pairs, but you can only go with a couple of millions, because those couple of millions already carry this information necessary to capture the full genetic variation, at least the common variation. So is this somewhat comprehensible, or we can try again? Okay, so and again, this is like what I explained. Like if we have this ancestral chromosomes, and the mutation is carried in only one of them. If we have many generations, then the region where this mutation, or this haploblock where this mutation lives, gets mixed with others, but it also gets very, very small. But in few generations, actually the mutation haploblock is very, very long, and that's why you don't need to have so many markers genotype to actually identify the causal haplotype and find the causal mutations. So this now brings us to the how it's possible that we are only using genotyping arrays that include a couple of hundreds of thousands variants, and then how we can then make it or infer it to be like 10 or 100 million variants, and how we can learn some biology. So this is again, it goes back like why the variants are linked. So to make it more graphic, here we have on the first 4 rows are just 4 different persons, and those are just like, let's say, like a chunk of genome that we have the full knowledge of their sequence. And here the letters in gray, those are the ones that are exactly the same across all individuals within a population, so it doesn't really matter who you look, it's always in this position it's A, and the next one is also A. And then there are certain positions that we call SNP or single nucleotide polymorphisms, which actually differ between individuals. And in genome, so this SNP that is considered to be at least frequency of 1% happens once per every roughly 1,000 base pairs. But again, it's based on the definition of your frequency. But there are certain areas or certain positions in the genomes that vary. And now if we just take all those, just consider only those SNPs that vary, we can actually construct those haplotypes that I was talking about. So this is, this can be considered as one of those million haplotypes that we have in the chromosome. And as I said, this one haplotype can have several versions. And here, for example, we have like, haplotype, 1 and 2 and 3 and 4 for the persons. But again, like if you start to like, although individually they are all independent or all different, but there are some, dependencies that, certain letters go always with certain letters. And then, although here you have like almost 25 letters to define a haplotype, you can actually come up or design or select only 3 SNPs so that the combinations of those 6 alleles actually, enable you to identify, which of the haplotypes you are currently dealing with. And that's why, although we started with, let's say, a couple of millions of base pairs, and we drill it down to 25, the ones that are varying or different between individuals, and then using some clever algorithm, we can actually only pick 3 SNPs that actually enable us to identify the full initial sequence. And this is again because of the linkage disequilibrium. And there was a question previously, what kind of SNPs or genetic variants are selected for a genotyping array, and this is the answer. There are selected only those that are informative for the genetic variation or this haplotypic variation. Or no, they also have like some genetically or clinically meaningful variants as well. This is how variants are selected for a genotyping array. But this, and now then you can use this, and this makes the genetic profiling very cost effective. The first genome, it took 10 years and more than $3 billion to sequence the first human genome. Because now the technology has developed, it only takes us, 8 days and $1,000 to sequence a full genome. But in order to like use those tag SNPs on a genotyping array, which has, let's say, 500,000 of SNPs, this only can cost as low as $20. And this actually makes it possible that we can, comprehensively characterize a human genome very cost efficiently and genotype many thousands or hundreds of thousands or even millions of people to actually do this type of genetic research we want to do, because the effect sizes are very small. And so, if we use those genotyping arrays, we can then, go backwards as well. We have then, genotype those tag SNPs. There exist, references with the different, haplotype contents and the full, human genome. So basically, then we can use only those couple of SNPs to actually infer the full sequence of the people that we are studying, and this process is called imputation. I don't know if someone is, commenting more on this imputation, but this is how it works. So we are predicting the haplotypes that we built initially using linkage disequilibrium. So this is just a brief snapshot how the machines look like that perform all the hard work of genotyping. So they are these days quite boring, black and white. Most thrilling part is actually this. If you put the machine working, it starts to go this Knight Rider, there is like no sound effects, but I'm still always into the lab. I'm wanting to put the sound effect there as well, But basically this is those are the machines, and those are in the bottom left. Those are the genotyping arrays. So this is if you remember from your high school, those the glass slides where you had the sliced onions or something like that on it. So this is exactly the size of this genotyping array, and this is the middle. This is the surface of this array. So there are this micro holes, and there are like beds where you actually have nucleotide oligotides on it, and it's again uses actually the basic biology from DNA replication to actually detect the SNP. So we know for every bed what type of SNP or what type of genetic position this is measuring, and it depends on if allele, let's say A or allele B, gets added. It's colored either red or green, and if for a certain SNP you have both alleles present in your DNA, it will be colored yellow. As you can see, all those different dots all correspond for a given a SNP that we're measuring out of those 500,000. And so then again, a computer program just reads all the different colors and then gives you the genotypes. So this is, as simple it is, the genotyping. Usually, genotyping one person, scanning, it takes an hour, but usually it's a process is like 2 days, the genotyping. And if we very briefly if we want to know the full human genome, not just the certain positions in the genome, so how it in principle work, not going into the details, we just take the genome, then cut it into the pieces randomly, and then we select that all the pieces are usually like 300 base pairs long, and then we just sequence randomly those 300 base pairs, and then we have, again by chance, the given position gets covered by many pieces, and then we again use computational algorithms to then put those very small 300 base pair sequences into one long sequence, and then this is how it looks like. So those are the different sequences, and although there are some holes here and there, on average you get quite even and nice coverage across the genome, and this is how we can sequence the full genome in less than 2 weeks, and this has a great impact on solving those Mendelian cases or Mendelian diseases I was explaining before. So over the past 5 years, I think almost half of the rare diseases have been solved. Prior to this technology, it was only a couple of hundreds or up to 1,000. So, but there are some other genetic variants as well, not just like a single nucleotide change, like either you have like this letter A, like adenosine or guanine, but there can also be like genetic changes where you actually have like repeative elements. Like here it's like a repeat of like 4 letters, and then it's like on certain haplotypes have like 5 alleles, some have 4 alleles, some have 10 alleles, and this is actually what is used. So it's called like short tandem repeats, and this is actually what is mostly used in forensics to actually identify who's the suspect, and again you don't need that many markers, and also fraternity is used this type of genetic alleles, and again how it says, like here is like the length ladder, and here like a control individual, here you have the victim with their specific bands, and then you have the suspects with their bands, and then for example also the sperm that was found in the crime place, and this is like also boyfriend mapped out, and then actually you can see that it's more than clear that actually the sperm that found in the crime site was actually coming from suspect 1, and not really anything to do with the boyfriend or suspect 2. So this is actually like although looking at all those crime TV series, very nice. They have like all this dimmed light and the spinning machines there and stuff. But usually it's just this kind of shell picture actually solves it, and it's less fancy in real life than they actually try to show it in the TV. But I think it's good to know like how this type of what type of information is used to in this type of situations. And of course there are other type of genetic variation is like how many copies of certain gene you have. You either normally is like you have 2 copies, but for example there is a deletion on your mother's side, or you have duplication on the mother's side, and this can again affect your phenotypic outcome. Mostly it affects cognitive abilities or mental retardation, and there can be also like even more traumatic changes, which is not just like a single genes gets duplicated or deleted, but then it's like a full chromosome, like for Down syndrome or for Turner syndrome, where the Y gets deleted. So it's just again like a background that it's the genome is way more complex and complicated and less understood than just the single nucleotide changes. So summary on the alleles in population. So large fraction of alleles have low allele frequencies, and as I mentioned, with low allele frequencies, it's very hard to actually achieve necessary sample sizes to get robust p-values to estimate if a certain change in the DNA is linked to a phenotypic outcome. Common alleles at the same time are shared between the populations, and that's also one of the reasons, as the common alleles are shared, and that's why it's also impossible to design genotyping arrays that can be used in many populations. Of course, genotyping array that is designed for Europeans only captures and in Europeans captures, let's say, more than 80% of the haplotypic variation, can only detect as little as 30% in Asia, and even less in Africa, and vice versa. If you design something that is like very predictive for Africans, may not really work for Europeans. And this is also one of the reasons why I think there was a question during the dinner why most of the genetic research now is done with European ancestry individuals. One is because there we have the large enough sample sizes, but the second and not that less important aspect is that actually the genotypic technology or arrays are currently designed for the Europeans. Of course, it's now moving on to use also different populations, but this is also one of the reasons why most of the research is done on the Europeans. Then another important point that genotypes and alleles are in equilibrium in infinitely large populations, but genes that are close together are linked, and this is how we can define haplotypes. And I will then show, in a way, I showed you how it helped us to comprehensively describe the genetic variation in the genome, but later on I will point out actually how it causes a headache if you want to interpret the biology using this type of data. Do we have questions about this part? Yes?
Just a question of haplotype, sort of like a variation. Do you have to have sort of a reference sample of people to sort of flesh out what is the haplotype variation and then do some kind of analysis on that? And if that's required, how does the population that you use, like if you don't have enough people in that initial sample, like maybe your chip is not as good. And sort of how is this developed?
Yeah. So, again, it may come as a surprise, but actually the initial arrays, and still the arrays mostly are designed on 60 European genomes. Okay, it also this sample also include 90 Asians and 90 Africans, but actually all this European content actually comes from 60 European genomes. So all this is called this HapMap2 consortia, and those HapMap2 imputed data. So this is 2.5 million variants. So this is all based on 60, all actually 180, 120 haplotypes they observed in the data, and that's why it's reasonably good for the common variants, like above 5%, but it's not really working that well in if you go in the lower frequencies, because it didn't capture this variance, and also as it's historically this 60 people come from actually from Netherlands, so this is actually like the variation it captures. But now we have like moved on. We have this 1,000 genomes reference panel. As I mentioned, we have there 2,500 individuals across 25 populations worldwide, and also the methodology where HapMap2 we had 2.5 million variants. In 1,000 genomes we have 80 million variants, and you can design your imputation. It doesn't matter if which population you do the genotyping in, because the reference population is so diverse. You can actually do a very good job in imputing also your population-specific variants if those are observed in the 1,000 genomes. But then I have, in the Estonians, we actually went further, and we have actually sequenced ourselves to 2,500 genomes for Estonians, and so selecting those to be, representing the different, clusters I showed you on the map of Estonia. So we selected people across Estonia, different counties, and then we constructed our own imputation panel. We don't really need to have our own array, although it would be even better. But already having our own reference haplotypes, we can actually do very, very good job on imputing alleles that are as low as 0.1%. Whereas in the HapMap2, you could do reasonably good job with alleles of 5%.
So it's great about the imputation process that makes it less important than the initial arrays.
Yeah, because in the arrays you just need to know, to in a way capture the genomic structure, like which variants go together with others. But actually, it's more for the imputation process to know exactly the haplotypes that you're looking, or the combinations of SNPs, how they sit in the haplotypes within your population. Then you can do a very, very good job in imputation. So these days it's more important to have your own population reference panel than your own population-specific genotyping array. Yes.
So there was some discussion, I think 2 days ago, about having different chips used for the same sample collection, and how that would require you to do sort of a meta-analysis across those 2 samples because of variation across chips. So I was wondering if you could talk a little bit about why that variation would exist and its effects on some of the analyses or results.
So I mean, you have the same individual's genotype with the different arrays. So actually, there, I would actually recommend that you try to, combine those 2 arrays together in one set. Like if you, for example, have your Illumina array, and then you have an Affymetrix array, and they have common only, let's say, a couple of 100,000, but then you have uncommon like many hundreds more. Then you can actually merge them together and impute them, and do an imputation. But and then have a very good imputation, because your input, this genotypes you put in for the imputation to infer the haplotypes are much more informative. But I think the other question can be, like, for example, you have a cohort, let's say 10,000, and then you have like 3,000 genotyped with one array, another 3,000 genotyped with another array, and then the third with the third array. And there actually, this is the case like what we have in Estonia. And there actually what I recommend is indeed you actually impute all the subsets separately, run the analysis separately, and then combine them into the, through the meta-analysis. Because across the different arrays, if you do the imputing, you have probabilities for the genotypes. And, in one array, something that can work very well doesn't really work on the other And if you just mechanically merge those three subsets together, and although still use some covariates for the array, it may not capture all this difference in the imputation accuracy. I don't know if this explained your...
Right, so I think it explained it, but I mean, the way that you presented it was that it's kind of very deterministic and the variance isn't as large as, you might suspect, but given the example, you say that it's large enough to be worried about it, right?
Yeah, so if you use those, generic association testing tools, like, the SNPs test, or I don't know what you're using here, they are actually the imputation uncertainty is taken into account. It's like you're weighted against, in your, association test. But, there you're not anymore, you cannot, wait for this uncertainty that efficiently anymore, because in some, the imputation has worked much better than for the other arrays, and there you can actually mess up your statistics. So this is why the variability comes in play. But again, discuss a little bit on later. So okay, now I have a little bit more on the regulation of gene expression, because like a gene expression is a way, like, you have as I said, like, there are roughly, I think, 20,000 genes, but not all are active all the time, and many are only active in certain steps in development, or certain tissues only, and can also be that in certain tissues the gene is expressed, or the mRNA is produced, like, say, 10 or 100 or 10,000 times more abundantly than in other tissues. So it's in a way, like, that regulates how gene may has a function, and why it's important, because I think an example of gene expression, we can in a way infer if the variant that we have identified in our association testing may be functional, or at least have some effect on the genes that are nearby. So this is just like again, very generic simplification of the process. So here we have something called the RNA polymerase. It actually consists of many hundreds of different proteins that form this complex. And what this complex does, it just binds where there is a gene. So again, there are some regulatory sequences that that call for certain proteins at first, and then a complex forms there, and then this complex starts to, like, unwind the DNA double helix. And again, based on this complementarity I explained before, where, like, always the guanine goes with cytosine, and adenine goes with thymine, and there's only difference that there is no thymine in the RNA, but it's uracil, but it's doesn't really matter. But again, as there are, like, these two strands, everything is very, very accurate, and RNA polymers also, although it's less accurate in the process than the DNA polymerase, it's still very, very accurate. And again, the structure of a gene, as I said, it's this is the promoter that initiates the transcription process, creating the mRNA. Then we have to, like, cut out the regulatory regions, like the introns, and then we have the splicing. And as I mentioned, like, the splicing can skip certain exons to create different proteins with a little bit different function, and there are also different ways how you can pack the protein together, and there are different modifications you can do with the protein. So it's either, like, has a longer life, or is active in a certain way. But this is, like, the basic structure of the gene. And as I mentioned, this is just, like, one example gene. It's like this complement 4A. It's like was a very big splash paper in Nature about how this gene is linked to schizophrenia. And here what we have is basically, we have, I think it's 400 people, and then from there is like from 40, I think, different tissues have been like sampled, and then the gene expression is measured, like, the abundance of this mRNA in the different tissues is measured. And the measuring the gene expression actually works quite similarly how I explained the genotyping working, where again use those fluorescent markers and then using, scanning those in and then estimating the intensity of the signal, and then you can measure the abundance of the RNA. And here, across the different tissues, here we have like liver, starting from the left, and adrenal glands and ovary, for example, and then we end with skeletal muscle and blood and lymphocytes. And there you can see that actually, and here we have a log scale, so you can see there it's like the gene expression in muscles is like 100 times less than actually in the liver. So, and this is just like, in adulthood, but it can be like in the different developmental stages, the gene expression pattern across different tissues is completely different. And, why or how the, as I said, like the gene expression across tissues is different, but, how then the genetic variation in your DNA actually also has effect on how the, like, within the tissue the gene expression can be different. Like here, for example, we have 3 donors, the different tissues colored like blue brain, red heart, and liver green, and then we collect their tissue sample, separate the RNA that are present, and then quantify the RNA present either using array technology or sequencing. And there you can see that the gene expression in heart tissue exactly the same across individuals and livers as well. But in the brain we see that in the donor 1 the gene expression is much more abundant than in the gene donor or donor 2 or 3. And then if we also have genotypes available for those individuals, we can actually see if there are any genetic variants that actually are associated with the abundance of the gene expression. And there indeed you see, like in the donor 1 has like GG genotype, and that's why the gene expression is very high, whereas the donor 3 has AA phenotype and the gene expression is very low. And how come, one may ask? As I explained, 95% or 97% all your GWAS signals actually fall within non-coding regions. So they are not in the genes, but they are regions that have some regulatory potential. And this is again a little bit like illustrate here on the bottom left. This is like it's like a regulatory sequence for a certain transcription regulator or like a gene expression regulator. So this binding motif for a given regulator, it's not like clear cut, but it's also like it has a higher affinity for certain combinations than others. And there, for example, let's say like this D and A here, it has a much higher affinity if there is a T allele or D nucleotide, and much lower where there is a allele. So in this case, in here, the certain regulator binds much more actively, and that's why much more RNA gets produced. Whereas the A, it still binds, but the binding affinity or the strength is not as strong, and that's why less RNA gets produced. And this is like a simplified figure showing that if for example here is the transcription start, like where the genes would start, there are some several regulatory regions before the genes, like something where the RNA polymerase itself binds, and some other helping proteins bind as well. So those are the regulatory regions that are quite close to the gene, and that's why. And later on, if we see how to define which is the causal gene, if you go for the nearest gene, you are usually almost correct, because the variants there somehow affect the gene expression, and that's why the closest gene may be the causal one. But there are also regulators for the gene expression that are far away and far away, like it's tens or hundreds of thousands of base pairs that may also affect the gene expression of a gene that is far away. Usually they are within the same haploblock. So this is like if all the regulatory sequence within one haploblock can affect one gene's gene expression. And that's also why sometimes you see that the variant lies within other genes intron, and although the closest gene, you want to say, oh, this is, although it's a gene's intron, it's like causal, but actually it's like the gene, let's say 25,000 base pairs upstream from this. And this is true for the lactose intolerance mutation. There actually the causal mutation lies in an intron that is, I think, 17,000 base pair upstream in other genes intron. So you would say that, oh, this is maybe the causal gene. No, it's something in the downstream. So but this shows like how in a way, like the gene expression could help, but I think it's also a good model for you to understand that sometimes it's much more complicated to say which is the causal gene using just the computational models. And very simply to explain, like what we are looking is like cis or local effects. As I mentioned, there is maybe a SNP in the regulatory region in the proximity of a gene, and then we can see a nice linear correlation between our genotype in the regulatory region and the resulting gene expression. But things go much more interesting if we, for example, look for trans or distant effects, and those are actually where, for example, we have a SNP in gene X, which is on chromosome, let's say 1, and this in a way affects the gene expression on gene on chromosome 2, either through directly changing like some proteins structure and binding affinity, and then this changed protein is not binding that efficiently anymore on the gene and regulating the gene expression, and then we actually are able to observe similar linear tendency. And this type of trans effects can either be like this, as I explained, that we directly somehow the transcription factor or the factor that regulates the gene expression gets changed, but it can also be like, as I mentioned, the chromosome areas in the unpacked nuclei, where chromosome 11 and chromosome 22 can be next to each other, and there can be some physical contact between the gene X and gene Y, like example of like gene-gene interactions. Or it can also be that there are some, for example, environmental factors. For example, this mutation or this SNP in gene A causes you to have higher levels of blood lipids or glucose, and then this feeds into the system spec, and then to this higher levels of glucose, your gene expression of Y gets changed. So it's quite complicated. So it's not just a predictive value of a polygenic score, but it's also the biology behind it. It's also quite thrilling. And again, just like one example for you to like this trans or distant effect. I think it's a very nice example on the prostate cancer. So there was a GWAS done on prostate cancer phenotype, reasonably powered, and then they found like this RS10187 424 on chromosome 2 is like strongly linked to risk of breast cancer, prostate cancer. And then, looking for, like, if this SNP specifically is affecting gene expression levels in genes nearby. There are 4 genes, and if you look at the predicted functions, you can, believe me that those are not very clearly linked to cancer etiology. But now we looked for trans effects, so if this SNP is affecting gene expression across the genome, and then what we found is that this SNP on chromosome 2 is actually affecting gene expression on like chromosome 1, 2, 6, 8, 12, 16, 17, 19, 20, and X. And if you look for functions of those genes, then those functions listed are much more obvious for the cancer biology, and provide several new biological leads to develop new drugs. So this is just like doesn't mean that all associations you find have such a global effect, but this is also another example like why it may be difficult to say what is the causal gene, or that the closest gene is just the one that is important. It as most of the variants are regulatory, they can have much more global effects. The epigenetics, as I mentioned, those are important in parent of origin effects or imprinting, and this basically epigenetics is how accessible is the DNA when it's active. So with methylation or those modifications on the histones, so the histones are the proteins that the DNA is packed onto. So if they are modified by methylation or the other processes, the DNA is not accessible for example, the RNA polymerase, and it cannot really bind to the DNA and produce mRNA for protein synthesis. So there are like several epigenetic modifications. I'm not really going into this, but there are like here we have like where we have the histones, and there are like a certain amino acids on those histones get modified with one type of the other molecule, and depending like where is the modification and what type of modification happens, it's either activates the center so it's accessible, or inactivates. So it's rather complicated. Yep.
On the last slide, when you said epigenetic changes are not heritable, what exactly did you mean by heritability in that context? Are you thinking about germline changes, or?
Yep.
Yeah.
So although there are controversial things about it, but epigenetic changes are considered to be not heritable. So like your epigenetic changes that happen to your to your parent are not transmitted to the child. Maybe there are some latest research on animals, but I don't know that in humans this definitely has not been shown that the epigenetic modifications are heritable.
So you don't necessarily mean heritability in the strict twin context of like comparing monozygotic and dizygotic twins and calculating heritability. You're sort of saying that like changes in the sort of germline are not directly translated from parent to offspring.
No, because I think in the process of fertilization of the egg, all those modifications get erased, and then new modifications get put on it. Yep.
But there are, like smoking.
Yeah, but this.
And smoking has epigenetic consequences, right?
Yeah, but I will discuss it on the next slide. But this is I just want to very nice example on having monozygotic twins, and this is like their the correlations on their epigenetic patterns from birth to the older age. And here we can see there are some marks that are highly correlated, and then actually there are marks that over time, are not correlated at all, and then there are marks that are not correlated in the beginning and are not at all correlated in the end. So in the twins, they, at birth they are rather similar, but as the methylation or this epigenetics actually can be considered as a marker of stress. So that it's a stress, and as Patrick pointed out, that that smoking is heritable, and smoking, and also there are epigenetic marks that are associated with smoking. It doesn't mean that the smoking, the epigenetic marks linked to smoking are also herited. And what is here the cause is that the smoking itself is a stress, and in response to the stress, the organism or the cells actually start to modify their DNA. So it's this epigenetic changes are a response to stress factors, and this is in here it's stress can be considered as age, or smoking, or overweight, and maybe there are some other examples, and it's also tumors. There's a lot of epigenetic changes, but those epigenetic changes only appear in the tissue of relevance to a certain condition. So, for example, if you are smoking, this has effect on your lungs and blood, so that's why you detect methylation marks linked to smoking in your blood. But looking blood-based epigenetic marks and linking it to childhood abuse, this doesn't really work, because this is not the tissue of action, and it's also there are not very good examples why it occurs. And then also, as a last point, is all those methylation or epigenetic marks. There are actually large fraction of the methylation differences between individuals are actually linked or associated with genetic changes. So I think half of the methylation differences can be actually explained by genetic differences between individuals. So again, then you can say that in a way this feeds into the this heritability that there you can measure, observe certain heritability, but is it actually the heritability of epigenetic marks, or it's actually the just clean-cut DNA-based heritability? Okay, so I think this is all for the gene expression and epigenetics. I think now we have a break for lunch. Is it true? And then if you want to have questions, happy to answer. And then on the last session will be about a little bit more on if you have run your GWAS and you have what to say about the biology. So thanks.